In this lecture, we focus on recombinant DNA technology, the techniques and biological systems that are used to study and exploit genetic material. Recombinant DNA technology employs the following components: a DNA source to provide a gene of interest, a cloning vector, which is a virus or plasmid which carries a DNA insert, a restriction endonuclease to make specific cuts in DNA, a ligase enzyme to allow the insertion of a DNA segment into a virus or plasmid, a host organism in which a plasmid or virus can be grown or amplified. Restriction endonucleases are hydrolytic enzymes which make cuts at distinct sites in DNA. The restriction sites contain a twofold axis of symmetry, that is, the sequences of the two DNA chains within the site are inverted, identical copies of each other. The positions where the restriction enzymes cut are identical for each chain, that is, they exhibit symmetry, and those positions can be staggered or even. In this example, 4 of the restriction sites have staggered cuts, which would leave sticky ends that can hybridize to each other. One of the restriction sites, HEY3, has an even cut, which would leave a blunt end. Note that restriction sites are sometimes called palindromes, suggesting that the sequences read the same in both directions. This is only true if the DNA sequences are read by crossing over at a central axis of symmetry. For example, the BAMH1 site reads GGA, and then you cross over AGG, and CCT cross over TCC. So the palindromes would be GGA, AGG, and CCT, TCC, but the palindromic sequence is on 2 different chains. Please get this straight, because you'll need to know it on an exam, and about 50% of the class screws this up every year. Here's perhaps the most well-known and often used restriction endonuclease, EcoR1. Notice again that the palindromic sequences occur on 2 different chains with a crossover point at the axis of symmetry. The endonuclease cleaves between G and A, giving a staggered cut with sticky ends of the 4 bases AATT. The utility of these sticky ends is that those ends will recognize each other by base pairing. Because of this, 2 different DNA samples, both prepared with EcoR1, will stick together at the ends and can be covalently joined through the use of a ligase enzyme. Digestion of DNA with restriction endonucleases produces characteristic fragments that can be separated using gel electrophoresis. The fragments migrate at a rate that's inversely proportional to the log of the number of base pairs. That is, smaller fragments migrate faster than larger fragments. In the example shown here, DNA from SV40 virus was cleaved with 3 different endonucleases. The 3 samples were then subjected to electrophoresis, and the gel stained with ethidium bromide. Southern blotting is a technique which uses a DNA probe with a sequence of interest to specifically hybridize to a DNA sample carrying a complementary sequence. The DNA sample is cleaved with a restriction endonuclease, subjected to electrophoresis, and the separated DNA fragments are transferred to a sheet of nitrocellulose paper. The sample is then hybridized to a radioactive DNA probe. The DNA probe carries a sequence complementary to a specific sequence in the DNA sample. Thus, the probe only binds to that specific sequence of DNA. The sample is then washed and placed in contact with X-ray film. The DNA sample band that's hybridized to the radioactive probe can be identified by its reaction with the X-ray film. Southern blotting can be used to detect genetic diseases that arise from mutations within genes. Mutations within restriction sites affect the size of restriction fragments, and this alters the electrophoretic migration of the DNA fragment. The detected mutation may either cause the disease directly or be closely linked to the mutation which causes the disease. The dideoxy method for sequencing DNA was developed by Dr. Fred Sanger. Sanger is also the first researcher to sequence a protein for which he received a Nobel Prize. In Sanger's case, lightning struck twice because he was awarded a second Nobel Prize for developing the dideoxy method. It's extremely rare to win 2 Nobel Prizes, but the double honor for Sanger was totally warranted. DNA sequencing experiments are conducted in vitro using the same components that are required to replicate DNA in living cells. Those components would be a DNA template, a primer molecule complementary to the template DNA with a 3' hydroxyl group to be added onto, 4 deoxynucleotide triphosphates, a DNA polymerase enzyme, magnesium, and several other protein cofactors. In addition to these components, there's an extra ingredient which makes sequencing possible. That's the dideoxynucleotide triphosphate. In a sequencing experiment, there are usually 4 reaction mixtures, each containing one specific dideoxynucleotide triphosphate. That would be dideoxy-ATP, dideoxy-GTP, dideoxy-TTP, and dideoxy-CTP. The presence of the appropriate amount of a single type of dideoxynucleotide triphosphate in the reaction mixtures causes DNA elongation to be terminated at positions where that particular nucleotide occurs. For example, if dideoxy-ATP were included in the reaction mixture, then some of the DNA would terminate at each position where an A occurs in the DNA sequence. So you would get a series of DNA pieces of different lengths, all of which terminated at the positions where A occurs. How does that happen? Stay tuned. This slide shows what happens when dideoxy-ATP is incorporated in a replication system. In such a system, deoxy-ATP represents about 98 to 99% of the total A trinucleotide, and dideoxy-ATP about 1 to 2%. So most of the time an A would be inserted in the sequence and replication would continue past that A position. However, about 1 to 2% of the new DNA chains would be terminated at each point where an A occurs, because replication cannot continue past the point where a dideoxy-A is inserted. You can see that dideoxy-A lacks hydroxyl groups at the 2' and 3' position on the sugar pentose ring. It's the missing hydroxyl at the 3' position which is unusual and which leads to chain termination. We'll come back to that shortly. You can also see that in the newly synthesized strands, some of the chains were terminated at the first A and some at the second. This pattern would continue for hundreds of nucleotides of DNA, giving a series of DNA fragments, all of which were terminated at the A position. When this experiment is repeated with each of the other 3 dideoxynucleotide triphosphates, a similar result is obtained. A series of DNA fragments would be produced which terminate at whatever N base was being used in the dideoxy-NTP. Now we're ready to look at the chemistry of the dideoxy method. The first slide again shows a primer sequence, CAC, with a free 3' hydroxyl group. There's an empty binding site for DTTP across from an adenosine residue on the template. In this case, a dideoxy-TTP replaces the normal DTTP. The dideoxy-TTP occupies the open site and hydrogen bonds with a complementary A residue on the template, resulting in TA base pairing. The 3' hydroxyl group with a primer carries out a nucleophilic attack on the phosphorus atom of the alpha phosphate of the dideoxy-TTP. That nucleophilic attack of the 3' hydroxyl group of the template forms a phosphodiester linkage with the alpha phosphate residue of the dideoxy-TTP and releases inorganic pyrophosphate. The DNA template or copy is now 1 nucleotide longer, and everything is ready for another round of DNA synthesis. The primer or copy now has the sequence CAC, and then there's that weird T. However, in this case, the normal 3' hydroxyl group on the T residue is missing, and there's now a hydrogen atom at the number 3 position. There's still an empty binding site for DATP across from a thymidine residue on the template. Now a DATP will diffuse in to occupy the open site. The A of the DATP will hydrogen bond with the complementary T residue of the template, resulting in AT base pairing. However, the 3' hydrogen atom of the primer can't carry out a nucleophilic attack on the phosphorus atom of the alpha phosphate of the DATP. Because the dideoxy residue cannot bond to the next DNTP, the process of elongation terminates at this point. Remember, this termination only occurs in a small fraction of the new DNA. For those rounds of synthesis where the dideoxy compound beats out the normal DNTP for the binding site, most of the DNA will continue to elongate until a dideoxy residue is incorporated. The sequencing method that we've just outlined, using 4 different dideoxy nucleotides in 4 different containers, works well for sequences of about 200 bases. However, that method has been improved so that the entire process can be carried out with 1 reaction mixture instead of 4. The newer method also allows for the sequencing of longer segments of DNA. The modified technique involves the use of 4 dideoxy NTPs, each of which has a different fluorescent dye attached to it. The compound shown here is an example of one of these dyes, in this case a dideoxy TTP derivative called di-TTP Big Dye Terminator, aka Mr. T to his friends. The compound consists of dideoxy TTP with a dye covalently attached on the side of the pyrimidine ring that's not involved in hydrogen bonding. This compound, like the underivatized dideoxy TTP, can pair with an A in the template DNA chain and cause chain termination of the new DNA strand. The fluorescence sequencing method employs the usual list of DNA synthesis ingredients: a DNA template, a primer molecule complementary to the template DNA with a 3' hydroxyl group to be added onto, 4 deoxynucleotide triphosphates, a DNA polymerase enzyme, magnesium, and several other protein factors. In addition to those normal components, there's an extra ingredient, the 4 fluorescent DNTP derivatives. Each of the derivatives fluoresces at a different wavelength so that each has a distinct signature or color by which it can be recognized. The elongation of the new segment of DNA stops when a dideoxy NTP derivative is inserted into the growing chain. The strands are separated using a capillary electrophoresis system from which the strands are eluted in the order of their size. The elution of the various strands is monitored with a fluorescent detector which can distinguish the 4 different fluorescent dyes. This figure shows the elution pattern from a fluorescent sequencing experiment. Each base has a different color code corresponding to a different fluorescent dideoxy NTP derivative. On this chart, a 360 base sequence is displayed. It is methodology such as this that made the Human Genome Project possible. This is the gene sequence of the bacteria Haemophilus influenza, which is the first free-living organism to be completely sequenced. That was accomplished in 1995. This table shows the history of genome sequencing. In 1996, the sequence of the yeast Saccharomyces cerevisiae was obtained. In 2000, the fruit fly genome was sequenced. Wow! Last but not least, a rough draft of the complete human genome was determined in 2001. The estimate of the total number of genes in a human is about 40,000. It appears that only about 3% of the human genome actually codes for functional genes. The other 97% of the genome may have some function, but the role of much of that extra material is unclear at this time. DNA fragments of known sequence can be synthesized using the solid-phase phosphite triester method. This procedure is especially useful in preparing DNA primers and probes for use in recombinant DNA studies. The first nucleotide is connected to a resin bead for ease of purification. That nucleotide has a free 5' hydroxyl group that undergoes reaction with the activated deoxynucleotide compound, deoxyribonucleoside 3' phosphoramidite. In the next step, the phosphorus atom is oxidized. Then a protecting group is removed from the new 5' hydroxyl group and the whole cycle can be repeated. In many cases, DNA material is available in very tiny amounts. Two examples where the DNA samples are often very small are in forensic criminal investigations or in biological experiments involving animal hair or scat samples. Many methods for characterizing DNA, including the sequencing methods that we have just considered, will not work with such small amounts of material. Before these methods can be used, the small DNA samples must be amplified to give larger amounts of material. The polymerase chain reaction, or PCR, is a method for amplifying small samples of DNA. About 20 years ago, Kary Mullis developed the PCR method, for which he was awarded the Nobel Prize in 1993. The PCR method is basically a DNA synthesis system similar to those we've been studying, with one important modification. In order to carry out PCR, a few DNA synthesis components are needed. A small sample of DNA to be amplified, 2 oligonucleotide primers, one for each of the 2 complementary DNA chains being replicated. The 2 primers bracket the region of the DNA to be characterized. If you have trouble visualizing this, look for the little yellow and purple regions of DNA on the next slide. Those are the primer regions. 4-deoxynucleotide triphosphates. Taq polymerase. This is the special component of PCR which allows the system to undergo repeated cycles of heating and cooling. It's a heat-stable DNA polymerase isolated from thermophilic bacteria which can withstand temperatures up to 95 degrees centigrade. The protocol for PCR involves 20 to 30 repeated cycles of heating and cooling. In the procedure, a duplex DNA sample is heated to 95 degrees centigrade for 15 seconds to separate the strands. The solution is cooled to 54 degrees centigrade, and short DNA primers are allowed to hybridize to each end of the gene. A solution is raised to 72 degrees centigrade, and Taq DNA polymerase is used to copy each strand. At that point, the cycle is repeated. The temperature is raised again to 95 degrees to achieve strand separation, etc., etc. The slide shows the synthesis of DNA in the first round of PCR. First, the 2 strands of template DNA are separated at high temperature. Then the temperature is lowered and the primers bind to a selected region on each strand. The synthesis of new DNA proceeds from 5' to 3' on both strands. Notice that in this first round, the new strands tend to run on beyond the primer location on the complementary template strand. As we've seen, the first round of PCR produces run-on copies of both strands. In round 2, the template and newly synthesized strands are separated from each other by a rise in temperature. The temperature is then lowered and the primers bind to each strand. The synthesis of new DNA proceeds from 5' to 3' on all 4 strands. Notice now something different is happening with respect to the run-on beyond the primer location. With complementary copies of the 2 original template strands, the new strands tend to run on again beyond the primer location on the template strand. However, the complementary copies of the products of the first round are terminated at the primer location. This happens because there's no DNA to serve as a template beyond this point. It turns out that with successive rounds of PCR, the short copies bounded by the primer locations tend to increase in a geometric fashion, whereas the run-on copies increase in a linear fashion. That means that many more short copies will be synthesized. In fact, if my math is correct, after 30 rounds, there will be 30 run-on copies of each template strand and millions of short copies that correspond to the target sequence. In round 3, the template and newly synthesized strands are again separated from each other by a rise in temperature. The process of annealing primers and replicating DNA is repeated. You can see that the short strands are starting to predominate. The figure shows how the cohesive N method is used to join diverse DNA strands. 2 DNA strands that are produced using the same restriction endonuclease can be joined together by annealing the DNA fragments with a DNA ligase. The cohesive or sticky ends will naturally adhere to each other by base pairing. The ligase enzyme then forges a covalent bond between the fragments. It's also possible to join DNA molecules which lack cohesive ends. The DNA fragments lacking cohesive ends, produced by mechanical shearing or other methods, can be given cohesive ends by the addition of a chemically synthesized linker using T4 ligase. The linkers are attached to the ends of the DNA fragments. Then the linkers are cleaved with a restriction endonuclease. Using this method, it's possible to add any restriction site to the end of a DNA fragment, and then clone that DNA into another fragment containing the same cohesive ends. Plasmids are small pieces of circular DNA that can replicate independently in a host organism. Plasmids are used to carry and amplify cloned fragments of DNA. The host bacteria can be grown for many generations. This allows the plasmid to be amplified billions of times. The plasmid shown here is PBR322. It carries an origin of replication, which lets it replicate autonomously in E. coli. It also contains genes for antibiotic resistance. One of those genes confers resistance to ampicillin and the other to tetracycline. When the plasmid is present in E. coli, the organism is resistant to those two antibiotics. In this diagram, recombinant technology is used to insert a sequence of foreign DNA into a plasmid vector. The original plasmid carries a gene for antibiotic resistance. When the plasmid is present in the host organism, an enzyme is produced that inactivates the antibiotic. As a result, the host organism becomes insensitive to that antibiotic. The isolated plasmid is cut open with a restriction endonuclease to produce a linear DNA product with two sticky ends. The cut plasmid is then mixed with a foreign fragment of DNA that has the same sticky ends. A ligase is added to the mixture and the foreign DNA is ligated into the plasmid. The recombinant plasmids are inserted into host organisms. When the host organisms are grown in a media containing the antibiotic, those organisms carrying the plasmid will survive because they're resistant. Organisms which do not have the plasmid will die because they're sensitive to the antibiotic. The surviving organisms can then be grown to amplify the plasmid, and then the plasmid can be isolated. Note that at this point, it's clear that the host organism contains a plasmid, but it might not contain the DNA insert. A plasmid that simply recircularized to its original form would also confer antibiotic resistance. In order to confirm that the DNA insert is present, other tests must be performed. When one attempts to ligate a piece of foreign DNA into a plasmid, there's always the possibility that the plasmid will simply close back into its original form with no foreign DNA inserted in it. To verify that there's indeed a DNA insert in a plasmid, it's common to use a plasmid containing a reporter gene. The reporter gene in this example is LAC-Z, which codes for an enzyme called beta-galactosidase. The LAC-Z gene has a restriction site within it that can be cut with an endonuclease. When the LAC-Z gene is cut and foreign DNA is inserted into the middle of it, the new product has a disrupted LAC-Z gene, which no longer codes for beta-galactosidase. If one selects plasmids which no longer code for beta-galactosidase, there's a strong possibility that the plasmid contains the insert. Here's a diagram showing the possible outcomes that we just outlined. The purple part of the plasmid represents DNA that comes from the plasmid, and the green part is the reporter gene that's been inserted into the plasmid. In the upper part of the diagram, the reporter gene has been cut with a restriction endonuclease. When one attempts to ligate the foreign DNA, coded in red, into the plasmid, some of the plasmids simply close up to the original form, shown on the left, while others gain the new segment of foreign DNA, on the right, in the middle of the reporter gene. The plasmids lacking the insert will express the reporter gene, whereas the recombinant plasmids with the foreign DNA will lack that gene expression. A simple enzyme assay will distinguish the two possibilities. This slide outlines a method for isolating a specific gene from the entire genome of an organism. This is a necessary preliminary step if one wishes to study a gene. Remember that the human genome contains about 40,000 genes, and those genes represent only about 3% of the entire amount of chromosomal DNA. In order to study a specific gene, it's necessary to insert that gene into a plasmid free of all the other genetic material in the cell. The first step to study a particular human gene would be to determine which of the 23 human chromosome pairs carry that gene. Having identified the relevant chromosome, one could then order up a genomic library made from that chromosome only. This library would be a set of plasmids with DNA inserts that span the entire chromosome. The library could be purchased as a mixed set of host bacteria containing plasmids having inserts that encompass the whole chromosome. The top of the diagram shows an agar plate containing individual bacterial colonies. That plate can be pressed onto a nitrocellulose filter, transferring cellular material onto the filter. Treatment of the filter with alkali causes cell lysis, and it fixes the denatured DNA from each colony to a particular spot on the filter. The nitrocellulose filter is then exposed to small radioactive probes. The probes are short segments of DNA which can hybridize to some known sequence of DNA in the gene of interest. In order to construct the probe, one must first know a small sequence of the gene. This gene sequence can be determined if the protein sequence is known or by other methods. The probe will stick only to those spots of DNA that contain the gene of interest. After washing to remove excess probe material, the filter is dried, then pressed to an X-ray film to make an autoradiogram. The spots on the X-ray that darken by radioactivity correspond to colonies carrying the target gene. One can then go back to the original master agar plate and pick those colonies having recombinant plasmids containing the gene of interest. Bacteriophage viruses can be used to clone large fragments of DNA that won't fit in a productive plasmid. The lambdaphage is often used for recombinant DNA studies. After infection with the lambdaphage, the viral DNA becomes inserted into the host chromosome. That's called the lysogenic pathway. The E. coli can then be grown for many generations, during which time the phage DNA becomes highly amplified. When sufficient cell mass has been produced, environmental changes can induce the expression of the dormant viral DNA. Under these conditions, called the lytic phase, many copies of the phage are produced and cell lysis occurs. The phage can then be collected and the DNA extracted. It's possible to cut out the middle of lambdaphage DNA and insert foreign DNA in its place. The product can be packaged in the virus and replicated in the host because it's the right size and contains all the necessary information for replication. Religated lambda DNA lacking the insert is too small to be packaged properly, and so it's not replicated. Genomic libraries that contain the whole genome of an organism have been produced in lambdaphage. These libraries can be used to isolate a gene of interest. The screening of a genomic library for a specific gene can be accomplished using hybridization with radio-labeled DNA probes, followed by autoradiography. DNA probes can be synthesized for a particular gene if a segment of the amino acid sequence of the protein is known. Because of the degeneracy of the genetic code, multiple probes must be used. With the 7 amino acid sequence shown here, 256 distinct probes must be generated to get the one that's the exact match for the gene of interest. Primer walking is a method for characterizing long regions of DNA. The first step is to isolate a DNA fragment with a known base sequence, which occurs at one end of the longer region. That region of DNA is then sequenced. At that point, a new probe can be synthesized which corresponds to the far end of the first DNA sequence. The new probe can then be used to characterize the DNA that's further along in the sequence. The process is repeated until the whole length of DNA has been sequenced. If a known messenger RNA can be isolated, it's possible to synthesize the corresponding double-stranded DNA, called cDNA. The process uses reverse transcriptase to synthesize a single DNA strand that's complementary to the messenger RNA. The messenger RNA is then digested away, and DNA polymerase is used to make duplex DNA. Retroviruses like the HIV virus have an RNA genome but copy it into DNA during infection. This DNA copy inserts into the host chromosome and behaves like a host gene. The enzyme reverse transcriptase is an unusual DNA polymerase that copies RNA into DNA. The host uses no such enzyme, making this a good target for inhibitors. These inhibitors are important because they can inhibit HIV virus replication. Reverse transcriptase uses the same basic mechanism employed by other DNA polymerases, except that the template strand is RNA. Nucleotides are added to the 3' hydroxyl end of a DNA primer. The 3' hydroxyl group attacks and forges a bond with the incoming deoxynucleotide. Inorganic pyrophosphate is released. The product of the first reaction now has a new 3' hydroxyl group, which can attack the next incoming deoxynucleotide triphosphate. It's possible to use cDNA in an E. coli expression system to produce messenger RNA and then to translate that messenger RNA into the corresponding protein. The cDNA is cloned into a plasmid that contains a highly active bacterial promoter upstream from the inserted cDNA. The plasmid is then transformed into E. coli, and the bacteria are grown and plated to give hundreds of single colonies. Those colonies that synthesize the desired protein can be identified by Western blotting using a radiolabeled antibody specific for the protein of interest. This method was used to clone the human insulin gene, the first recombinant gene to be used for a medical purpose. The Western blotting procedure uses specific antibodies to identify proteins of interest. To carry out Western blotting, E. coli is infected with the plasmid carrying the gene of interest. Individual bacterial colonies are grown up on an agar plate. The bacterial colonies are then lysed to expose the protein. The protein spots are transferred to a nitrocellulose filter. A radiolabeled antibody specific for the protein of interest is added. The filter is placed on X-ray film, and the dark spots on the film are used to identify the colonies containing the protein of interest. DNA microarrays, also called gene chips, can be used to simultaneously analyze hundreds of genes from different sources. Here are 1,733 genes from 84 breast tumors being analyzed simultaneously. The pattern of gene expression for an individual gives clinicians information about the type of tumor and the mode of treatment. DNA microarrays are used in molecular biology. They consist of an arrayed series of thousands of microscopic spots of DNA oligonucleotides. The spots of DNA, which are called features, each contain picomoles, that is 10 to the minus 12th moles, of a specific DNA sequence, and are known as probes or reporters. These probes can be a short section of a gene or other DNA element that's used to hybridize to cDNA or cRNA samples called a target under high stringency conditions. Probe target hybridization is detected using fluorophore, silver, or chemiluminescent labeled targets to determine the relative abundance of each target. Since an array can contain tens of thousands of probes, a microarray experiment can accomplish many genetic tests in parallel and thus can dramatically accelerate many types of investigations. DNA microarrays can be used to measure changes in expression levels to detect single nucleotide polymorphisms, or SNPs, or to genotype or resequence mutant genomes. Site-specific mutagenesis can change one base or a number of bases at a specific site in a gene. With oligonucleotide-directed mutagenesis, a primer containing a single mismatched nucleotide is used to produce a desired single change in the DNA sequence. Cassette mutagenesis is used to introduce a synthetic oligonucleotide into a gene using restriction endonuclease cleavage and ligation of a specific altered nucleotide sequence. The inserted sequence can include one or more base changes. It can also include deletions or insertions. This series of photographs were all taken around or on the University of Minnesota campus. Here's the iconic Weissman Art Gallery at sunset. Here's another view of the Weissman at sunset. I just can't get enough of these shots. Here's a close-up photo of the Weissman taken again at sunset when the building displays a fantastic array of colors. How many different shapes can you see in this photograph? For a couple of years, it seems workers were constructing an